they get his opinion, then a different course of action might happen. The same thing on the right, whether it's Carmela or his son Anthony Jr., or his daughter Meadow, or even his physician, his psychiatrist Dr. Melfi, or even his uncle who's his consigliere, might need to give him information on a need-to-know basis, but also to make the final decision will be him. And so this is kind of the way I look at the immune system. This is, in reality, we joke about the Sopranos, but in reality, this is the way the immune system functions, let's say in an infection. So in this case, this is Plasmodium falciparum infection. And what happens when protozoa get loose into the bloodstream? And so you've got, parasites here. I'm trying to get here we go. Parasites that are binding to, the endothelium. And there are different chemicals that are being released by erythrocytes, in this case, nitric oxide. But then there's other things that happen locally, whether that be macrophages who are, taking in opsonized complexes with, antibody and also complement. And then certain cytokines are being released, right? And then B cells are being activated. And then from there, they're starting to make either IgM or IgG, and then a Th1 response is starting to be initiated. On the other side, you've got dendritic cells, and again, more Th cells, T regulatory cells, NK cells, iNKTs, gamma delta T cells, again, more macrophages and other cells, and more cytokines, and then actual symptoms like fever, and whether that be hypotension, afterload reduction, things like this. So again, this just demonstrates, this is just the merozoite part of the infection of Plasmodium falciparum, and it demonstrates the complexity and also what it takes to defeat a pathogen, and that in reality the odds are against you, and that's why in ID we rely on antimicrobials and other medications to kind of help with this. Any questions? So there's different ways to look at this, okay? There's obviously a gross anatomy, there's a histological anatomy, and then there's a molecular anatomy. And in this case, this is the gross anatomy, and this is taken from the Cleveland Clinic. Of all the different parts of the immune system that are located throughout the body, you can see in green representing, the lymphatic system with lymph nodes and lymphatics that drain eventually into the venous system before it enters the right atrium of the heart. These lymph nodes are a sentinel place for B cell development and macrophage activation, T cell development as well, and also a way for pathogens to get through the body too. You also see the spleen there, which holds up to 20 to 30% of all of your red blood cells and platelets, but again is also a way for the immune system to start to regulate on infectious processes that are systemic. Then you have the thymus gland, which is just located just in anterior to the heart, and this is where up until you're about 20 to 21, depending on the patient, where T cells mature, and then eventually go into the lymphatic system and throughout the rest of the body. But up until that time, your thymus gland is activated and allowing T cells to develop and or if not to be destroyed if they are too reactive to your own cells. Bone marrow is part of the immune system as well, and this is whether it's in the long bones or the larger bones in the body, and this is where most hematopoiesis and myelopoiesis transpires, right? And so and again these cells get into the rest of the circulation and into the lymphatics, and here we are. But I do want you to also notice that the intestines are here. And what people forget to realize, and you probably remember this from step 1, and as I tell the fellows, stuff on step that you memorize for step 1 comes back to bite you here in ID because it's critically important. And again, about 70% of your entire immune cell repertoire is in the intestines and also in the respiratory tract. This is located in areas called lamina propria, which are just just below the epithelial layers, and also areas called Peyer's patches, which are, very large nodes, quote unquote. Basically, it's like a lymph node, but inside the intestinal wall. And this allows and we'll talk about this in a few minutes. This allows the cells of the intestine and the immune system locally to figure out if there's a friend or foe in the GI tract or respiratory tract, and thereby alerting the rest of the body of what's transpiring. So again, basics. There are going to be some new concepts for you, but a lot of this should be reviewed. Okay? So, how did this all start? Well, even simple forms of life develop the basic forms of immunity. Things like, even sponges, for goodness sakes, have innate forms of immunity that then, little by little by little, were, tracked up into, more sophisticated invertebrates and vertebrates. But all of this started essentially with one-celled organisms, and then slowly but surely went up the chain. So, pathogen induction is a big deal. And, a lot of things happen when, let's say you eat something that is infected with a pathogen, let's say norovirus. There's different types of things that are activated. It's not just, oh, your immune system, gets activated and here we are. It's much more complex than that. And I'm going to try to, make this as painless as possible. But there are things like PAMPS and DAMPS, and we'll talk about this, and in terms of how the immune cells and other cells in the body recognize friend or foe, or when one of their cells that are part of the body gets injured, and what happens there. So tell me the difference between innate and adaptive, somebody. What does that mean? Anybody?
Innate, you're born with it.
Innate means you're born with it. Yes, very good. And what does adaptive mean in the immunological sense?
You're exposed to something and develop a immune response to it.
Yeah, to put it a little more sophisticated, you encounter something, realize that it's bad, and then you do something about it, and then you remember it, right? And then sometimes you have to stop yourself from, doing something about that all the time, and that is called, effector cell, memory cell, and regulatory cell. And all those are parts of the adaptive immune system. There are some cells which cross that path and, kind of teeter on both the innate and adaptive sides of things. We'll go through that, but that's essentially what it is. So innate cells are more, built around not, of course, their cells, but also things like complement, innate cytokines, antimicrobial peptides, all through the recognition of different PAMPs and DAMPs. Okay? And adaptive immunity is kind of what I described. Much more sophisticated, seen in higher vertebrates, where you've got different types of lymphocytes, whether that be B cell, T cell, that have an effector, phenotype, a memory phenotype, and also regulatory phenotype. So that then, you can do something about a pathogen, remember that pathogen for the next time, but also not go overboard and cause collateral damage because of said pathogen. So, like somebody eloquently said, innate immunity is what is native or what you're born with. Okay, and this is the initial form of immunity that you're born with, while the adaptive immunity begins to prime throughout life. And it's much more than just cells and some funky molecules. It actually has to do with also anatomical and physiologic barriers. So, name me some of these anatomical or physiologic barriers.
Your
Not all at
skin,
once.
saliva,
Skin,
the ciliary.
saliva. Excellent. What else? What else?
We have the lymphatic system, of course.
Lymphatic system, I would say, is probably more attuned to the adaptive immunity.
Right. Sure.
If you think about it, let's say you are, some kind of crustacean. They don't really have that kind of broad immunity. However, they have certain things that help them, right? An exoskeleton, right? They also have other things protecting their brain, right? Like a blood-brain barrier. That is critical, okay? What else?
Mucosa.
Okay, mucosa. So what happens with that? Okay, let's say you go outside and it's February or March here in Florida, and there's a lot of pollen in the air, and you start having, your nose becomes wet. What is that, right? And you start coughing. Those are physiologic barriers, right? So a cough, a sneeze, things like that are, are ways that our body tries to expel things that are irritating the body locally, right? So the same thing with why people, why you have diarrhea with certain illnesses, right? The body is trying to eliminate the pathogen, whether that be because of toxin, or whether that be because it's just, stimulating the epithelial lining and M cells, causing a smooth muscle reaction, and that's why it happens, right? So it's much more apparent than we realize as physicians, and that there are things that, or like, you scratching after a mosquito bite. Same, all these things kind of go along with that, right? Give me some molecular defenses.
Like IgA?
So IgA is kind of a initial, innate response. However, it's a little more on the adaptive side, and the reason is because, again, some innate cells have to recognize, what they're seeing as a pathogen or as something that could be a pathogen, and then refer it to the B cells, whether it be in the lamina propria, Peyer's patches, other places. You do secrete some native IgA, but it usually occurs after, some type of antigen presentation. So what else?
Complement system.
Complement system, excellent. What else? Come on, guys, this is going to be a long presentation. What's in tears that is very important?
Lysozyme.
Lysozyme. I heard the good Dr. John say that. Very voice. Yes, lysozyme. What else? Who here eats honey with whether it be in dessert or their tea or coffee? None of you? Okay. So even things that are inside things like, in honey, there are antimicrobial peptides that your body generates, whether in the liver or whether that be locally. Different types of cells make their own too, which combat infection. And they actually bind certain types of carbohydrates, lipid structures, protein structures on the surface of the pathogen, and then alert the rest of the immune system that there's something wrong. Cellular defenses are obvious, right? We're talking about phagocytes mainly, and other cells which kind of tell the adaptive immunity what's going on. The other thing that the innate system does is presentation of antigen, which, again, like I just said, also activates the adaptive system. And it can also retain memory as well. There's a regulation of the initial immune response, basically putting a brake on things, so the train doesn't completely go off the rails. And there's also a cleanup crew. So what do I mean by that? So again, more Sopranos kind of references. It's after there's a contest and combat between the pathogen and the immune system locally. There's some collateral damage, and some cells are too injured to survive, or they're apoptotic. And so then some of these innate cells clean things up. This is what I'm talking about, right? So, and a couple of things that we didn't mention. So we're talking about here on the left, right? anatomical and physiologic barriers, intact skin, lysozyme, tears, also those low stomach pH, right? And ciliary clearance inside of the lung. The innate immunity, right? Eosinophils, macrophages. Mast cells we're not going to cover today, but just to know that they're there. Neutrophils, dendritic cells, and then notice that they're natural killer T cells that are on that border between innate and adaptive immunity. Okay? And then things at the bottom, right? Talking about mannose binding lectin, things like dectin-1, that can identify different types of fungi, let's say, by recognizing certain carbohydrates in the surface of an organism. Complement, LPS binding proteins, C-reactive protein, antimicrobial peptides. Then on the other side, the adaptive immunity, we're talking about B cells, T cells, natural killer T cells, and also antibodies. Let's talk about the complement cascade. So this is an ancient form of innate immunity. It's, again, in lower invertebrates and vertebrates, of course. In humans, there are small proteins that are produced in the liver. And so when the liver is damaged, there are issues with complement. Or if there is a rheumatologic disease where complement is consumed, right? And certain tests, mainly fungal tests, are based on a complement antibody binding, thus opsonization, leading towards opsonization. There are also precursors that are released from the liver. They're activated by either whether it be chemokines, cytokines, and then, depending on where they're at, different proteases will cleave them and they'll become active. So there are three different pathways that are part of the complement cascade, all right? And this includes the classical pathway, the lectin-mannose pathway, which is, again, is a recognition of different types of carbohydrates, and the alternative pathway, which, again, all end up in the same exact thing, which is, and we'll see this on the next slide, destruction of the pathogen by either opsonization, the inactivation of the immune system systemically, or creation of what's called the MAC complex, which destroys gram-negatives. This also can be, complement can also be part of, demonstrating that a cell's infected with a virus, and also fungi. And so it's not just bacteria, but also protozoa. And I can tell you from personal research that it's really important in the pathogenesis and clearance of Plasmodium. So this is taken from Nature Reviews in Room, and you can see the three different pathways, right? So, and they're all numbered. Well, most of them are numbered, or they have a letter component. And so, like I said, there's the classical pathway, which is, there's some antibody that is bound to pathogen, and then starts to bind the C1 complex, merges with C4 and C2 to create this this protease, which cleaves C3 into two different components. And one stimulates the different types of immune cells, mainly T cells and B cells. And the other one then, of course, binds to antibody and goes for, and then this way leads to opsonization. The lectin pathway binds to a carbohydrate. In this case, it could be mannose, but there are other carbohydrates that can be bound. And there's other proteins that are then bound to, and then it goes through the same cascade. The alternative pathway really is more of a cycle with C3, and it stimulates, factor B, factor D, and then leads down to the same kind of cascade, which eventually leads to C5, which can also stimulate cells, but then also create this MAC complex, which destroys mainly gram-negatives. And there are certain immunological deficiencies in the C5 to C9 complex, which can make you prone to certain gram-negative infections, in not just Neisseria, but also others. Questions before I move on? So next are antimicrobial peptides. So these have been known for just only a short period of time in terms of the medical literature, probably about the last 10, 15 years or so. They're found in higher organisms, both invertebrates and vertebrates. And the classic, thing that we actually found out was that honey is loaded with these antimicrobial peptides. If you notice, honey really, it's really hard for honey to spoil, okay? Whether that be for fungi, whether it be for bacteria, right? I mean, it is a nutrient-rich food, right? That's why bears love it, and, and so do we. But um, it's this halogenation of one of this of specific antimicrobial peptide, which causes it to have antibacterial properties and also antifungal properties. In fact, and I will digress, honey is actually used in different parts of the world for different types of skin ulcers, including diabetic foot ulcers, as the mainstay because of these antimicrobial peptide properties. It is actually used in different places around the world where there is an enormous amount of, whether that be Madura foot, Charcot foot, and other ailments of the skin. They target bacteria on the vast majority, but also again, fungi, viruses, parasites, and even dysplastic host cells. So cancer also can stimulate antimicrobial peptide formation. Okay, and these antimicrobial peptides usually occur locally. They're no, it's not relegated to the liver per se. But again, if you have liver dysfunction and therefore do not have a proper production of protein for the body, then the antimicrobial peptides can suffer. Okay, there's two different mechanisms of action. Usually, there's a cationic to anionic bond, which causes a lysis of the organism. It causes basically a depolarization and then a destruction. Okay. And then there's an intracellular modulation of certain cellular processes, whether that be DNA synthesis, translation, even transcription. And that then will eventually lead to apoptosis and subsequent lysis, right? And also this stimulates various innate immunological processes. Thereby, it's part of the first line of defense for certain pathogens. And this is exactly what I kind of just said, right? So these peptides, which are short, they're short, amino acids, anywhere from 30 to 50 amino acids in length. Okay, they're not a full, fully functioning protein per se, but they do like what I said here, whether that's a cationic, anionic bonding, destruction of the cell, then, or inhibiting it, cellular function intracellularly and causing lysis. But it also does all kinds of cool things on the other side, whether that be stimulation of dendritic cells and neutrophils, the monocytic lineage, and T cells, and even B cells, and will start to bind things like LPS and other things in the body, including free RNA and DNA after cell lysis, which then will allow certain other mechanisms to be stimulated, or in some cases regulated on. This is also a novel, interesting area for auto inflammation in the body, and is being studied in the rheumatologic and allergy fields. Questions? Okay, let's move on. So what are PAMPS? So no, it's not short for Pampers, but PAMPS are actually what are termed pathogen-associated molecular pattern molecules. Okay, and by the definition, here from this paper in 2015, they're a diverse set of microbial molecules, okay, or antimicrobial molecules, that then share a number of different genetic or general patterns, rather, or structures that alert immune cells to destroying intruding pathogens. So if an E. coli is blown up, right, you have LPS and other genetic material, let's say other proteins that are considered foreign. These are then detected by the immune system in different ways, right? And whether that be through this pattern recognition system, which are famously a good example is like toll-like receptors, or other domain-like receptors, which can then alert the immune system that there is a pathogen present, and then that can start the ball rolling so that you do not get sick. This is part of the problem with why LPS in large amounts causes such a problem by reducing afterload and generating such a robust innate cytokine response. It's because of these PAMPs. And so again, every organism has it. So this is why you're able to recognize some of these things. But again, it's just like everything else. If the immune system is not really primed for look for these things, then they won't be, and that's why you can have a problem and succumb to an infection. So what are DAMPs? So these are damage-associated molecular patterns. So these are actually endogenous molecules, right? They're derived from the host. And so they're sensed by different innate immune receptors. So basically, they're the PAMPS of your own cells. And what happens? So let's say you have a cell that is dysplastic and not really functioning well, and start to engage in itself in, apoptosis, and that cell dies and explodes. Okay, fragments of that cell are going to, be exposed locally to the immune system. The immune system then starts to think, uh-oh, this could be, a cancer locally, and so the beginning of a cancer locally, and thus we have to do something about this. It also could be for, viral infected cells, but it also can revolve around autoimmune diseases like neurodegenerative diseases and things like this. And so it's much more complex than that. So again, it could be, from damaged or dying cells, and again, promoting a sterile inflammation where it might not be an infection per se, but it's anything that's the body does not recognize as, homeostasis. The whole key here is to return to par, okay? And so it's to get back to homeostasis. And so what kind of examples of this? So this HMGB1, okay, is one thing. But then other things like nucleotides and ATP, if they're released from a cell, that shouldn't just be hanging around. And so the immune system recognizes this, and then it will subsequently take this under consideration and start looking for a culprit of why that cell died. Questions? Okay, so this is a nice cartoon of what I kind of just described. So at the top, a pathogen, let's say in this case a virus, releases PAMPS, and then is subsequently recognized by whether it be a dendritic cell or a macrophage or something else, whether it's through this pattern recognition receptor like TLR and other types of receptors, and then we'll stimulate T cells to start looking for a culprit, all right? And then what happens then, right? So you've got cytokines and chemokines that are generated. It might actually generate a regulatory response and might not initiate an effector response. So, and malaria is a good example of that. But there also could be things like tissue repair, right? And a tempering of the adaptive immunity, like I mentioned. On the bottom are things, like DAMPs, where a cell is stressed, will release these, these molecules, like I mentioned before, and they're picked up by the same types of cells, and then relayed to T cells to kind of figure out what's going on. So the way that I like how Vallis and et al. Kind of described this in this paper from 2014, where they said that PAMPs are like identification of strangers, and DAMPs are identification of dangers. So I think that's pretty cool, right? Where you can, there's things that kind of tell you that there's a bad guy around, or if one of your people is hurt, and then what are you going to do about it? Questions? Do I still have everybody, or is everybody asleep?
No, we're with you. Keep going.
Okay.
Yeah, we like this stranger danger.
I like that. I like that, Christian. Stranger danger. I knew that you would be the one who would appreciate that the most. So I digress, and I'm just kidding. So let's talk about phagocytosis, right? So, this is it's the prime way for your body to figure out what's happening around it at the histologic level, right? So the definition is basic process by which a cell, it doesn't matter which, ingests through a process called endocytosis and internalizes another cell, parts of cell, or even particulate matters, and then decides on what to do with that information. And there are two types of phagocytes, basically. The ones that are the professional phagocytes, right? These are the guys who, this is what they do all day long, okay? And who are they? Talk about neutrophils. Eosinophils can actually be phagocytic. They don't really phagocyte other bacteria or protozoa or helminths per se, but they will phagocytose particulate matter and decide on what to do, whether that's to release granules or not, or even communicate with other parts of the adaptive immune system. Okay, monocytes in their lineage. So who's that? So we're talking about free macrophages, dendritic cells, resident macrophages, right? Like Kupffer cells, osteoclasts, microglia. Yes, all these are parts of the monocyte lineage, and their job is to protect, the cells in their area by sampling everything around them to see what's going on. Then there are non-professional phagocytes. These are cells that don't do this as their job per se, but they do it to sense what's going on and then refer back to the immune system. So who are we talking about here? We're talking about fibroblasts, epithelial cells, endothelium. They all do it. And so they can process debris or parts of cells, these are these damps that then go and relayed back to the immune system per se. So what about lymphocytes? So do they phagocyte stuff? Of course they do. So B cells can, would be rare, but the ones who really do are different types of T cells, mainly these natural killer T cells, things like gamma delta T cells and stuff. I could do an entire lecture just on these cells, but again, you have to, you got to crawl before you walk. So before we get there, we really have to go through the other processes first. But just know that there's types of T cells that are on that border between innate and adaptive immunity that can phagocytose different types of debris and cells to decide on what to do with it. So again, we're talking about the professional phagocytes here, right? So their main focus is to clear pathogen locally from an area of the body. What else do they do? So secondary focus is to opsonize antibody complement pathogen complexes and remove them from the bloodstream. And where is that done? That's done in places like the spleen, and the liver, and even the bone marrow. Okay, so all these are done in these types of areas, so that then if there is a pathogen in significant amounts, they know what to do with it and get rid of it. Okay. Tertiary focus is to present antigen to the T helper cells, okay, CD4, via this MHC complex, right? So the major histocompatibility complex 2. There's also type 1. We'll get to that in the next set of lectures. But this is the main way of how these phagocytes and things like dendritic cells present antigen to T cells and B cells and let them know what's going on. Kind of like going up the chain. Then there's a quaternary focus. And this is what I was talking about before, whether it be to clear an area of damaged cells or apoptotic cells, and by these damps and the signaling through their through the same mechanism. Any questions? So let's go on neutrophils. So we're talking about their granulocytes in origin. They're stimulated by IL-8, causing them to leave the bloodstream and enter an area where their services are needed. They're actually the most common type of white blood cell. And so they phagocyte pathogens. Doesn't have to be bacteria-wise, but it could be fungi and other things. They also can stimulate lymphatic T cell responses locally. And then they release nets. What are nets? Anybody? Huh? Come on, guys. This is supposed to be interactive. I have no one? No one's on the call with me? Hello?
They're neutrophil extracellular traps.
Okay, so the good Dr. Tony here kind of went through and helped you guys out. So what are nets? So yes, they are a complex of basically, the neutrophil becomes a sea cucumber and unleashes its guts, quote unquote, okay, where that be different protein and DNA complexes that then are extruded either from a dying neutrophil or from a neutrophil that's still alive but is about to die, and basically throws it out locally. And this creates, a net basically, and that ensnares different pathogens, takes them out of causing more damage locally, and then from there, reducing the burden locally of that pathogen and allowing the rest of the immune system to compensate, right? So this happens in, let's say, pus. It's dead and dying neutrophils, but it's also composed of, what's inside those neutrophils that then allow them to snare a pathogen. In this case, let's say it's staph or Pseudomonas or something, and then reel them in so that then other phagocytes like macrophages and the like can actually destroy them, and also the apoptotic neutrophils as well. So this is a really cool concept. And again, when somebody's neutropenic, they lose this ability. And so it's not just the fact that you lose your Pac-Men, you're also losing this ability as well, which is pretty cool. Okay. Questions?
Is that just chromatin, or is there more to it?
So again, it could be, it's chromatin, DNA. It might just be a DNA fragment. There's protein. There's all kinds of other components to this. It's actually still an evolving part of the literature right now. It really depends on the type of cell or actually the bacteria or fungus that neutrophil is encountering. But you're right. It's for the most part a mix of protein and chromatin/DNA.
Thank you.
So eosinophils, again, I wouldn't call them a professional phagocyte per se, but they still do their job to determine what's happening locally. So again, they're part of the granulocyte lineage. They're stimulated by IL-5, a little different cytokine, okay? And they release cytotoxic granules towards protozoa and helminths, okay? But they also phagocytosis, remnants of these pathogens, okay? And they thereby stimulate lymphocyte T cell responses. So, are they Pac-Men per se? No, not necessarily. However, they can take in, different things, whether it be PAMPS or DAMPs, so that they can figure out what's happening locally. Macrophages. Okay, so these are the, besides neutrophils, these are the kings of phagocytosis in the body, right? So they're monocyte lineage. Now, the way I was taught this, and this is, I'm imparting this to you because it might be interesting, but basically macrophages are amoeba that don't divide. And so everything that amoeba can do in the, in the environment a macrophage can do as well, except they just don't divide. That's why in a disease like cytomegalovirus, it invades macrophages and replicates there, but the macrophage doesn't divide. So what happens? It becomes enlarged. Hence the name, giant cell, and then, cytomegalovirus, right? So they reside in all parts of the body, right? Liver, spleen, CNS, lung, dermis, GI tract, whether they be lamina propria, Peyer's patches, bone. They can also be a Trojan horse carrier of pathogens. Now, this is the last time I'm going to bug you guys. Give me some examples of this. This is critical.
Mycobacteria.
Mycobacteria, excellent. What else?
Tropharema.
Tropharema, excellent. What else?
Yersinia pestis.
Yes, so Yersinia could do it. Less likely, but it can do it. What about things like Listeria, Legionella, okay, Histoplasma, right? Leishmania can do it. Plasmodium can do it, right? These are all Brucella can do it for sure. These are all pathogens that Coxiella can do it. These are all pathogens that Salmonella, right? These are all pathogens that can hang out inside of macrophages and have a latent stage, which then can attack later on. So that's the downside of these cells. This is that because they have this amoeba-like feature that they can be a Trojan horse for different, So what do? They eat stuff, sure, whether it be through just the entire organism itself, or bound to complement an antibody, but also they can do antigen presentation. Okay, and they're potent producers of cytokines, whether that be an inflammatory response, okay, that's the MAC1, or the MAC2, which is more of a regulatory response. And sometimes, if they sense that they're in trouble with, let's say, one of these Trojan horse pathogens, they will engage in apoptosis, and then DAMPs will be released, and then the immune system will be alerted of a problem locally. This is a nice cartoon of a macrophage and the different things that it can do, whether it be antimicrobial actions, like producing different oxide radicals, antigen presentation, the antigen antibody uptake, like the FC receptor, and to stimulating growth factors for wound healing, phagocytosis, and of course, like an osteoclast, bone resorption. Dendritic cells are a cousin of macrophages, but they're a little different. Okay, they too are part of the monocyte lineage, but they're similar to macrophages. However, besides just removing a pathogen from the area, they're really good at antigen presentation. That's really their function. And so, they're the ones who are in constant contact with T cells and B cells to figure out what's happening locally. Now, yeah, they're ubiquitous throughout the body, just like macrophages. However, in areas where the antigen detection is really important, like GI tract, respiratory tract, GU tract, they're in higher numbers. So again, their shtick is really antigen presentation more than anything else. And just like with macrophages, they too have a pre-stimulatory phase, and then either an effective phenotype or a regulatory phenotype is generated. And this is exactly what I'm talking about. We have a mature dendritic cell at the top of this cartoon, but you see that cytokines are generated, in this case, IL-12, 18, IL-5, IL-15, and different type 1 type 1 interferons, and things like IL-1 through IL-18, whether it be through NK cells, whether and different types of T cells, or even the halting of destroying every single cell in a location just because one cell is infected with a virus. And there's also stimulation of B cells as well. So this is a complex diagram, and there's a lot of different things that can go on here. But again, it really depends on the type of pathogen that's encountered by the dendritic cell and what is happening. So non-professional phagocytes. So what is this? So these are epithelial cells, endothelial cells, fibroblasts, and even microfold cells, also known as M cells, which are in the GI tract and also parts of the respiratory tract. And so they sample, particles locally through endocytosis, figuring out what's friend or foe. Okay, and they will let whether that's NK cells, other macrophages that are local, innate B cells, innate T cells, including iNKTs, and dendritic cells on what's happening locally. And this is all done through MHC1. So again, M cells are really important, let's say, for something like salmonellosis, which is one of the first cells that phagocytoses the organism and then brings it in, and then subsequent macrophages and dendritic cells are infected and then causing an infection locally. But this is a way for, again, your immune system to kind of figure out what's going on, right? Questions? So briefly, even though we'll talk about this next time, it's important to talk about natural killer cells, okay? So they're lymphocytes, but they're a little different. They, like iNKTs, are part of that innate immune system, right? So they respond quickly to a wide variety of pathological challenges, PAMPS and DAMPs, and this is why they're really important in figuring out if a cell is infected with a virus or an intracellular bacteria or fungus, and even detection of dysplastic cells leading to cancer. So they bind to MHC1 and then determine that course of action depending on what the antigen that is presented is in this case. And they also kind of play a role in regulation of T cell response. So again, they can pump the brake to figure out what's going on if that needs to be done. So this again is a little cartoon about this from Science. And again, whether that's a stress-induced response or a cell that is infected, NK cells will figure out what's going on. They'll either blow up the cell or that through use of granzyme and porphyrin and other things, and then they'll stimulate cytokines to either stimulate T cells or macrophages or other things, and they will also represent antigen should that need to be done. And so in this case, you see the green is stimulatory, whether that's interferon gamma, IL-1, and then the regulatory IL-10, IL-13, those kinds of things can occur where they'll pump the brake of the immune system. So how does this help how does the innate immunity help by antibiotics? So those of you who have taken thus far have been in the ASP course with me and Dr. Tony at the VA. You know this is the first thing I tell you, right? It's a numbers game. So you have to achieve the appropriate kill of pathogen and thus reduce numbers. So what is that? So with different types of infections, you can have different loads of pathogen. And the immune system can only handle so much. And so if the immune system is overwhelmed, well, then the pathogen has the run of the body and can either kill the host or damage it significantly. The immune system can't have that. So what did we do? Antibiotics are actually bioterrorism agents for these pathogens. And they help weaken or kill the pathogen, and thereby reducing the numbers and allowing the immune system to have a fighting advantage, right? And that's really what it is. It's the return to par, return to homeostasis, right? But like, when you guys have discussed antibiotics in other courses and with me on ASP, that's a double-edged sword, right? We've got adverse effects, pathogen resistance and whatnot. But that's the true, reason why antibiotics are used. It's the immune system, like Alberto said earlier, that really helps, clear the infection and leads back to homeostasis. The antibiotics just make it easier for them. And this is a good example, right? So antibiotics, again, create dysbiosis in the microbiota locally, stimulating innate immunity, adaptive immunity, and innate immunity will also stimulate adaptive immunity. And then subsequently will stimulate memory that way, and your immune system is primed for the next pathogen next time this comes around. So then you might not need antibiotics. So next time. So it's almost that time, so we'll kind of conclude. So we'll talk about adaptive immunity, right? Which is a much, much bigger discussion and much more complex. We'll talk about B cells and T cells, and what they do. We'll also talk about innate T cell immunity, which is a specific interest of mine, actually. And I think, and what my hypothesis is, how different types of infections, escape the immune system, and through, whether that's modification of parenchyma of an organ, and how that changes the innate T cell immunity, causing an infection to become chronic or relapsing. We'll talk about different chemokine, cytokine responses, and how that influences the following: naive cell function, effector cell function, memory cell function, regulatory cell function, and how that plays into removing pathogens from the circulation or from a place locally, and how your body stays sharp for the next time, and also how it not to create damage locally because of this. And also, how to identify what pathogens could be infecting your patient just by knowing where there's a problem in the immune system. You can figure out almost 99% of the time what that pathogen or what pathogens could be causing this problem in the face of an immunologic syndromic diagnosis. And I know I can see in my mind the second years rolling their eyes at this, but it's true. Again, understanding that syndromic diagnosis, understanding what parts of the immune system are going down and what you could be seeing because of that. So next time, we will have more Soprano stuff, and understand that, yeah, Tony Soprano is in charge, but he can't do it without his capos and other guys on the ground to get things done, to make sure that everything is safe in the neighborhood. So thank you very much. Any questions?